Biotechnology and agriculture. Agriculture biotechnology applies to not all technologies used on the farm. It usually involves biological or chemical processes. It includes a broad category involving many different types of technology and science. It consists of breeding, genetics, microbiome research, synthetic chemistry, and animal health. For example, biotechnology has reduced the use of costly inputs and has been noticed to improve weed management, allowing farmers to reduce tillage for better soil, water, and air quality. Technology is usually considered synonymous with the biomedical research, but there are many other industries that take advantage of the biotech methods for studying, cloning, and altering genes. Throughout life, we have become accustomed to the idea of enzymes in our everyday lives. When looking at agriculture and biotechnology, it is more than just fighting disease or improving the quality of food. Despite the rapid take on biotechnology by farmer and strong scientific agreement that biotechnology does not pose on health and environmental risk, burdens are slowing the research and innovation of new biotech traits and are starting to reduce U.S. farmers' international competitive advantage. And did you know that roughly 90% of corn, cotton, and soybeans grown in the United States have been improved through biotechnology? Gene editing in biotechnology is very important throughout agriculture biotechnology. It has been studied since the early 1980s, and gene editing is the ability to make highly specific changes in the DNA sequence of a living organism, essentially changing its genetic makeup. The development of gene editing technology for gene therapy, however, proved difficult. Much early focused on not correcting gene mistakes in the DNA, but rather on attempting to minimize their consequence by providing a functional copy of the mutated gene. Genetic engineering and enzyme optimization techniques are being used to develop better quality feedstocks. There are 4 different common methods of gene editing: CRISPR, TALENs, ZFNS, and homing endonucleases or meganucleases. CRISPR stands for Clustered Regulatory Interspaced Short Palindromic Repeat and is used to change DNA sequences and gene function. Gene editing and genetic modification are often thought to be the same, but they are different in little ways. GMOs often use foreign DNA, whereas gene editing is used to make more precise changes to plants' own DNA. But many of the methods are labor-intensive and inaccurate. The CRISPR/Cas9 system has become a very popular tool in gene editing due to its high accuracy and ease of use. The significant leap in gene editing tools brought new urgency to long-standing discussions about the ethical and social implications surrounding the genetic engineering of humans. Gene editing to treat disease or to change one's traits dates back to at least the 1950s and the discovery of the double helix, which is a structure of DNA. Molecular mistakes were the fundamental ideas behind gene therapy, and from the 1980s was seen as the holy grail in molecular genetics. Dr. William M. Muir, a biotechnologist at Purdue University, stated, "If you imagine our genome as an encyclopedia, then gene editing is the ability to change words, sentences, punctuation, or paragraphs." GMOs are genetically modified organisms. A GMO is an organism that has had its DNA altered or modified in some way through genetic engineering. In most cases, GMOs have been altered with DNA from another organism, sometimes referred to as a transgenic organism. According to the USDA, GMOs are used to plant over 90% of all corn, cotton, and soybeans grown in the United States, which means that many of the foods you eat likely contain GMOs. While some people claim GMOs are harmful to your health and environment, notable organizations and research say that GMO foods are safe and sustainable. In the food industry, the GMO crop genes added them for many different reasons, as in improving their growth, nutritional content, sustainability, pest resistance, and ease of farming. GMO crops are incredibly common in the United States. GMO crops and farming make it much easier, but there is still concern about their potential effect on the environment and their safety of consumption of humans. The Food and Drug Administration, Environmental Protection Agency, and the United States Department of Agriculture claim that GMOs are safe for human and animal consumption. Genetically modified foods offer several advantages to the grower and the consumer. Crops have been modified to have genes that protects them from pests and insects. An analysis of 147 studies from 2014 found that GMO technology has reduced chemical pesticide use by 37% and increased crop yields by 22%. The previously listed factors help lower the cost for the farmers and consumers. It allows greater crop yield and growth through harsher conditions. Genetic modifications can increase the nutritional value of foods and may be used to enhance the flavor and appearance of some food. But there is still a concern about the long-term safety and environmental impacts. And since cancer is caused from DNA mutations, people fear eating foods with GMOs because it may make the cancer progress. And the American Cancer Society has stated that there is no evidence to link GMO food intake to an increased or decreased risk of cancer. When looking for GMOs in your food, avoid them by limiting GMO ingredients, eating locally, looking for third-party non-GMO labels, or eating and buying 100% organic. The term cloning can describe many different processes that duplicate biological material, such as natural twinning, reproductive cloning, and therapeutic cloning using sperm cells. In early cloning, the theories that led to embryo splitting was stated in the late 19th century. The background of cloning tells that reproductive cloning is reproduction without fertilization, which means it's asexual. Identical twins, triplets, and quadruplets is a result of spontaneous natural split of the embryo. In humans, about 1 in 80 births are the result of a twin pregnancy, although the rate of twinning varies genetically among ethnic groups. There are numerous things that can complicate cloning, but there are no complications that go along with cloning alone. One thing that can go wrong is the embryo may fail to develop during the in vitro stage. In vitro is a medical study that is in a laboratory within the confines of a test tube. Another risk is LOS, which is large offspring syndrome, and it's seen in pregnancies of cattle and sheep that come from cloning. Most clones that are normal at birth become as strong and healthy as any other young animals. Clones that come from calves and lambs tend to have more health problems at birth and may be seen as more likely to die right after birth than conventionally bred animals. Results from extensive health records from research institutions show that the clones are perfectly healthy and walk, wean, grow, mature, and behave just like conventionally bred animals. Biotechnology is a very important part of agriculture. It is used in many different situations and should be learned and taught thoroughly and correct.